Just glad to be here, just to let you know. Oh. And we have worked on lipid binding selectivity to the G6W amyloid dodecamer. As you can see, we're interested in G6W, and what G6W is G6W is the peptide that forms the double helix dodecamer sculpture, which has a hydrophobic core plasma for lipid binding, and it also is a mutation of KV11. And if you don't know what a KV11 is a peptide of a chaperone protein of an alpha-beta crystalline. Today we are interested in G6W is because it is a decamer, which it is a large oligomer as well, and come to find out that it's more toxic than KV11, and it has a cavity of potential lipid binding. As you can see here, you can see these are sculptures of G6W. As you can see for the front one, you can see the G6W, and you can see the cavity right here. As you can see at the bottom, this is G6W as well. It just shows you a better inside of G6W. As you can see, those decamers as well, and that palmitic acid is binding into the cavity of G6W. And the overall reason why we're doing this research is to see if the double helix sculpture can act as a lipid binding protein. Our hypothesis. We hypothesize that G6W has the ability to accommodate the alkyl change of fatty acids or lipids due to the size and shape of its cavity. Additionally, we hypothesize the length and saturation of alkyl change affect the binding and ligand to the G6W dodecamer. So our main material is our instrument again, which is called the ion mobility spectrometry mass spectrometry. I'll be explaining you how does our instrument works again. So, because it's very important. As you can see, the electric field will pull those ions into that entrance funnel. As those ions enters that entrance funnel, it will then enters into the drill cell, which is separated by the shape and size. As it enters out that drill cell and exit funnel, it will enters inside an ion guide, which that ion guide will push those ions throughout that quadricle mass filter, and it will separate by the separate by the mass to charge ratio. Now going over to the collision cell, we will not be using because the collision cell it breaks it up into fragments, which we want our sample to be intact, so we will not be using the collision cell for our experiment. For the detector, it detects the ions to make the spectrum, and time of flight mass filter, it shows the resolution of the peaks, which you can see the peaks will show sharp and narrow and not flat. Give you a better insight of our instrument, I have these two scenarios, same species but at different conformations. As you can see, these ions, the blue one, since it's the biggest, it will have a difficult time throughout that drill cell, which it will proceed through it slowly than these small ones. The green one will be the easiest one going throughout that drill cell, which it will proceed through that drill cell much faster. Now going over to our scenario 2, oligomers at a different charge state. You have to know is that the greater the oligomer size is, it will go through the drill cell much faster due to the higher charge state. That being said, since the trimer is the highest, since it's a charge state of 3, it will go through the drill cell much faster than a dimer, which is a charge state of 2, and a monomer, which is a charge state of 1. And what's so unique about our instrument is the drill cell, which I highlighted, which it shows different species at the same to mass charge ratio. Going over to our lipids, I will be explaining you what are these lipids and why are they different and why are we using these lipids. As you can see for POPC, we are using POPCs because it is a common lipid. As you can see for the other 4, we are using those because it is a PC head group and it has a chain of variations of POPC. And why are they different? It's because POPC, it has a 1 saturated on top, it has a 1 unsaturated at the bottom. As you can see, it has a double bond as well, which is a 16 carbon chain. As you see for DPPC, it is a 2 saturated with no double bonds, and it is a 16 carbon chain. Where it's 16,1PC, it is a 2 unsaturated, it has double bonds, and it is a 16 carbon chain as well. DSP and DOPC, they are both 18 carbon chains, which one doesn't have double bonds and one does. Which you can see for DSPC, it is a 2 saturated with no double bonds. and it is an 18 carbon chain. And DOPC, you can see, is a 2 unsaturated with double bonds, and it is an 18 carbon chain. Going over to our results, like I said before, if you don't know how our instrument works, you will not be able to know or see our results and get the information. As you can see for POPC, it had a moderate amount of binding lipid going on. You can see that the intensity was kind of low, but it did pretty well. It wasn't the best sample that we done. As you can see for DPPC, it was very weak, but the intensity was very low as well, but it wasn't the worst sample that we done. 16-1-PC, it was very strong, as you can see. The intensity was very high. DSPC was weak as well, which was the worst one that I done during that sample, during all my samples. As you can see for DOPC, it was very high, just alike as 16-1-PC, which the intensity was pretty high as well. And as you can see, the master charge, and you can see the intensity as well. And you may ask, what are these right here? And these are just showing you how G6W is being added to the lipid complex. And that being said, 16-1-PC and DOPC was the best ones that did pretty well with lipid binding. But out of all of them, 16-1-PC would be the best because it has a higher intensity, as you can see, which you can see it has 10 to the 5th, and you see DOP has 10 to the 4th. And I didn't really have to zoom into 16-1-PC than I had to do for DOPC. Going over to my conclusions, lipids containing unsaturated alkyl change binds better to G6W dodecamer, which you can see 16-1-PC and DOPC, they are both unsaturated. Both 16 carbon chain and 18 carbon chain unsaturated alkyl change bind to G6W. As you can see, 16-1-PC is 16 carbon chain, and the 18 carbon chain is a DOPC, and they're both unsaturated again. And you can see that it binds pretty well on the right side to G6W. And we have a method to compare binding of lipid ligand to amyloid oligomers. And you may ask, how did I get this conclusion? And how did I get this conclusion is because of the MS peak. It shows you that it helps you determine the abundance of a lipid peptide complexes. And these are my citations. And I just want to say thank you to the SMART program for giving me the opportunity to be here today. And I also want to thank Dr. Tan and his graduate students, Sarah and Alex, and all his graduate students for giving me the opportunity to be here and be in their lab for the summer. And I also want to thank Dr. Jackson, Dr. Brothers, Dr. Ellemeyer, Dr. Rio, Dr. Aina, and my family and friends, because without them believing in me, I wouldn't be here today. Thank you.